First of all, I'd like to say that I'm extremely excited about this meeting. It's in a sense, it's not exactly a culmination, but a very necessary step in our maturation as a community in terms of conservation policy. And that may sound a little bit strange, but I hope to explain to you why I think that's the case. We as a scientific community, as a group of people interested in this particular field, I think it's fair to say, perhaps with the exception of North America because of your legislative framework, have very much lagged behind many of the other disciplines within conservation biology in terms of how we've translated our research into policy relevant information and how we've managed to get our lexicon into the general discussions in conservation. I think this meeting that we're going to have over the next couple of days is really going to help. I hope very much is really going to help. As already has been touched upon by several people, it's going to be far from easy. If I would like to define a role for myself over the next few days, it would be that I'm going to have to be the bad cop some of the time, because I think for many of us in scientific research, pure academic research, it sort of seems to us that we have to be extremely of course, very rigorous in what we do, very, very considerate of all of the different variations in the parameter space that we're interested in exploring in defining indicators. But ultimately, the politicians don't care about any of that. They're interested in a few very key, very simple indicators that will allow us to incorporate genetics into biodiversity trend analysis. And that's an uncomfortable truth sometimes for us, because it means that we have to let go to some extent of some of the complexity that we are normally used to dealing with and embrace, in fact, in our work. So I'm not going to say much more than that at the moment, but what I'm going to do actually is give you very much the sort of policy perspective that I've developed, especially actually from rather not being involved in the North American system, which of course, where conservation policy and the role of genetics is much, much more active. With the Endangered Species Act and the COSIWIC in Canada, you are the only countries in the world that have bespoke legislation or guidelines in using genetics in conservation management. And the rest of us, frankly, are sort of working in the dark in comparison. But what that means is that it's been a very interesting thing for me to watch the way that it's developed, the debate has developed over the years, but also it has really made it very important for us to get organized outside of North America. And I'll sort of come on a little bit to that. I'm not going to talk about this slide very much, except to say that, to reiterate that the US and Canada has a long and distinguished history in the incorporation of genetic data in actual endangered species legislation. And that's actually in sharp contrast in many ways to what's occurred elsewhere. For example, if we look at the EC Habitats Directive, which was first written in '77, but the major version is in '92, uses the word genetics once and only in the context of population connectivity. So it's a very, very different situation indeed. This is very much in terms of understanding wild genetic diversity. What I'm going to focus on a little bit is from 2010 onwards, and really some issues that were really that are very important. The most important one being the Convention on Biological Diversity's HE targets for 2020. But also one very, very important paper, cri de coeur really, by Linda Leiker and colleagues, that was published, which basically, it's self-explanatory, neglect of genetic diversity and implementation of the Convention on Biological Diversity. And also in Europe, the formation of the first group really trying to explain genetic diversity to policy and managers. But I'll talk a little bit about IPBES and other things as well. So I think that the thing that the game changer for us was, in many ways, was this rather surprising HE target that came out from Nagoya in 2010, which is target 13, which is very explicit. By 2020, the genetic diversity of cultivated plants, farmed and domesticated animals, their wild relatives, but also, and this is really important for those of us who are interested in wild species, other socioeconomically as well as culturally valuable species is maintained, and strategies have been developed and implemented for minimizing genetic erosion and safeguarding genetic diversity. So that's a key game changer really in terms of genetics. Obviously, the most important thing to remember is that every country that's a signatory of the CBD is free to pursue its own interpretation of this and to institute its own programs that are pursuant of it in the way that best suits its interests. And I'll come back a little bit to that. But I think there are two terms that I want us to remember and talk a little bit about over the coming days. First of all, genetic erosion. So it'd be really, really interesting to get everybody's opinions and ideas on what that term actually means. Okay, I think where it came from, having talked to the people that were in the room when this was incorporated into the targets, but I don't think that the people in the room that included this necessarily thought about it in all its dimensionality. And then safeguarding genetic diversity. What are we aiming to do? What does safeguarding mean? And I'd like to come back to that later. I'm not offering any opinion on that at the moment, but I just want to put it out there for this particular juncture. And as I said, Sean's already shown this wonderful, logo, which basically says that we failed for the 2010 year of biodiversity, failed all our targets, so we'll change it into a decade instead, and we'll fail again probably. But all the different countries that are signatories to this have all developed their own national strategies. Genetics is much more present in some of those national strategies than it is in others. And it's done the way that it maps on to other things like the HE targets varies extremely. So for example, the French National Biodiversity Strategy is extremely clear, but if you look at the categories of targets that they've been set in their own biodiversity strategy, the map onto the HE targets is not necessarily always the same. Indicators, Sean mentioned indicators, and I think this is one of the things that I really wanted to talk a little bit about, and it be partly because we've had a somewhat bruising relationship with the indicators. The CBD is reliant on, and I just don't want to bombard you with acronyms, but is reliant on an organization called SBSTA, which is the subsidiary body on science of technical and technological information. And SBSTA is responsible for commissioning all sorts of conservation organizations to produce indicators. One of the biggest organizations involved in this is called the Biodiversity Indicators Partnership, or the BIP. They produce all sorts of wonderful products, including an iPhone app called the HE Targets Passport, which is supposed to tell you a little bit about our progress in reaching the HE targets. But also BIP gives funds for doing necessary development of indicators. So for example, Sean and myself applied to BIP last year to develop a wild species genetic indicator, and they came back to us and they said, you're nowhere near there yet. And that was a very, very disappointing thing, but I think they were absolutely right. So, we are as a community a long way away from translating our thoughts on important parameters and important things to measure in genetic diversity and converting them into an indicator. Here are some indicators, and these are that you go on the BIP website. And here's one of them, which is important, which is trends in genetic diversity of species. And there are two current prevailing indicators for trends of genetic diversity of species I would like to add of all life on Earth. And they are the numbers, the diversity of ex situ crop collections, and the genetic diversity of terrestrial domesticated animals. The genetic diversity of terrestrial domesticated animals is not measured in the way that we may consider that be. So it's not measured using molecular genetic diversity. It's about the number and distribution of livestock breeds. Okay? So these are incredibly important, of course, for humankind and for sustainable development. They map onto the Sustainable Development Goals very well. They're very important indicators, but I think it might be important for us all to agree right now that they are not the full story of genetic diversity of life on this planet. And that is one of the key charges that we have, is to try and move ourselves along so that we can also be represented in addition to domesticated genetic diversity. And so this is the Global Biodiversity Outlook, which is one of its goals is to promote the conservation of genetic diversity. And this basically says the same thing as the AG target. Progress has been made towards conserving genetic diversity ex situ, but here are some other, while the genetic diversity of wild species is more difficult to ascertain, the overall decline of biodiversity of this presented in this report strongly suggests that genetic diversity is not being maintained. That's important because what it's saying is that people are currently using species diversity and population size, things like the Living Planet Index, as a proxy for genetic diversity. Now, we may all be able to agree that in some cases that's going to be a good fit, but probably not in all. And this is coming back to what Sean was talking about, state pressures responses, and this is part of the biodiversity indicators approach. What's happening basically in terms of populations of biodiversity? What are the pressures that are affecting that biodiversity, and how are they changing over time? And how is biodiversity actually responding, largely through management and direct interventive conservation measures? Okay, and if we look at again looking at a whole variety of different indicators out there, but here you can see indicator F, genetic diversity of terrestrial and domesticated animals. This is over time. We can see that in general that genetic diversity of terrestrial domestic animals is declining and is predicted to decline even further towards 2020. And basically, even with these general indicators that we're looking at here, as you're probably aware, the midterm review of the Aichi targets that was presented in November 2014 was not at all optimistic that we would be able to achieve many of the targets that were set. Currently, indicators are being further developed by an organization called ARTEG. And again, I don't want to bombard you with acronyms. This is the Ad Hoc Technical Expert Group for the CBD Science Body. And ARTEG have been discussing in further indicators development. This is the latest version of their indicators assessment, which happened about a year ago. And here's target 13 again. And these are the indicators that they've been talking about: trends in genetic diversity of cultivated plants, domesticated animals, trends in extinction risk and populations of wild relatives, trends in protected area coverage of wild relatives. And this is really maps back on what Sean was talking about in key biodiversity areas, although only in this case with reference to wild relatives. But here's a big blank part of the table: trends in genetic diversity of socioeconomically as well as culturally valuable species. In other words, wild species that matter. We could argue that many of them do. So this is completely blank at the moment. And trends in development and implementation of strategy for minimizing genetic erosion and safeguarding genetic diversity. And they're really only talking about genetic resources for food and agriculture. So it's these two big areas of Target 3 indicator development that indicate just exactly what a vacuum, in many ways, we are operating in and that we need to fill. This is where Arteg thinks that we are today, okay? It's basically an indication of whether or not an indicator is available or under active development. Again, you know, there are some blanks here, and so it's really, and there's not even, you know, necessarily organizations that are stepping forward to work on this. So this is where we think we are today, and it's clear that there are large areas where there's not an awful lot of activity going on. And most importantly, this is where Arteg thinks there are gaps, and trends in genetic diversity of socioeconomically as well as culturally valuable species is a large gap. One of the points I would make is I would argue that, in fact, the way that the current indicators work for domesticated species as well is not adequate. It's just measuring, you know, ex situ accessions effectively. And the only indicator that Arteg thinks that is ready to use in a genetic context at the moment is the trends in extinction risk and populations of wild relatives, and that's not even a genetic indicator at all. So that's where we really are at the moment. One of the other factors and global activities that's forcing greater sort of sight into genetic diversity at the moment is IPBES, the Intergovernmental Platform on Biodiversity and Ecosystem Services, and the role of biodiversity and ecosystem services forces obviously us to look at all levels of biodiversity, including genetic diversity. And there's a mapping across exercise between what's happening in the Aichi targets in the CBD, but also this global 2018 global assessment of biodiversity and ecosystem services building. And part of the issue around that is that the role to which genetic diversity potentially underpins provisioning ecosystem services is something that is a real key and active discussion that's going on at the moment. And you know, we could talk about this a lot, but understanding ecosystem resistance and resilience and the difference between those, in other words, the ability of the state of a particular population to be maintained regardless of the anthropogenic or other environmental pressures on it, so population size doesn't change, or the resilience of a population, in other words, a population that is hit by an environmental issue, for instance, a pressure that forces it into an alternative state, but has still enough genetic diversity and demographic viability to be able to bounce back into its former state. And at what point does that reverse direction change become impossible? And of course, we all of us, I'm sure many of us are interested in showing and talking to politicians about that. But we're still, and this is just one set of important examples on the role of genetic diversity in resistance and resilience. But there are still actually rather few examples out there of the direct either experiments or very strong evidence on the role of genetic diversity in resistance and resilience. And in an IPBES context, that is really, really important for us as well. But I would like for us to think around this kind of resistance resilience concept as well, because that is going to map much more explicitly onto the IPBES requirements. And slowly but surely, the role of genetics is beginning to take on a little bit more visibility. This is a Giabon paper that Dan and myself and several others were involved in from a few years ago, where we actually managed to get allelic diversity as an EBV example on the essential biodiversity paper, not necessarily proposing that or even drilling down into what that actually means, but at least it's starting to be mentioned, it's there, and people recognize that it's potentially important. So these, the roles of indicators are going to be really, really important. They need potentially to assess condition, so the state, to potentially provide an early warning of change, to be sensitive, but not too sensitive obviously, to other variation, and if possible, diagnose the cause of the change as well. But there's clearly going to be trade-offs that we're going to have to make between these desirable features and what's available, and the whole question of how many different parameters we will look at, and how many of those can be aggregated or need to be disaggregated into different indicators is also important. They need to be easily measured. We need to be able to do this everywhere, easily, cheaply, in a relatively broad way. They need to be easy to understand, relevant to population management. They also need, in principle, to respond to stress or whatever we want to measure, changes in population size, in a relatively predictable manner, and thus also have high sensitivity and, in principle, low variability in their response. So, again, in an ideal sense, they need to be anticipatory, signaling an impending change in a system. So not necessarily only diagnosing a small population paradigm, but a declining population paradigm at the beginning of the decline. So act as an early warning system. So, equally, they need to be have an observable response before we get into a red flag situation, enable us to simulate and potentially predict ways in which we can change things by our actions, and have a known response to natural disturbances and anthropogenic stress. And I didn't put papers up, but there are lots of papers out there now that show the difference in genetic diversity in populations that are under anthropogenic modification versus not. And they need to be integrative. So no pressure, it should be all very straightforward. So yes. In terms of their mathematical properties, the indicators should, in principle, be able to change in a proportional way. They should be able to give values that mean the same in different species. In other words, we need to think about interoperability among different species within the same ecosystem, for example. And they need to be something that is effectively relatively impervious to the populations that we're looking at, the time scale and the spatial scale as well. So these are things that already start to raise ideas in my mind, at least, as to the sort of genetic parameters that we can be looking at. But of course, we all know that, as Sean has already said, we know that they're not met by many of the standard statistics that we use, the markers that we use, where there are huge differences in power with the different marker approaches that are being taken and the way they're being applied, and then how they're being statistically analyzed, how we summarize the data, which are the best ways to summarize the data, and also what they actually mean in terms of fitness. So, and viability of populations. The correlations between molecular diversity and fitness in many cases are very, very low indeed. And also going back to what I was talking about in terms of resilience, the issue around complicated performance of statistics that are there to examine changes in population size. So those are the problems and challenges. I'll very, very quickly, have I got, yeah, okay. As I said, as a community, we've been trying to get the message out there outside of North America to the other communities, and particularly in Europe, we've got this project called Congress, which is a forum between policymakers and geneticists, where we often talk about genetic issues. We've got a very live one at the moment on the Scandinavian wolf that's being discussed a lot. And it includes a bunch of very simple stuff. So we have knowledge packs. We have, as I say, we have a forum of engagement. We have a simple publications database that links to Google Scholar, because most policymakers don't have access to most journals. We have a beautiful decision-making tool that was designed by Sean, and a number of other facilities as well that we're trying to build the group, and a bunch of very simple things. So we actually have a PowerPoint presentation that is free for anybody to use, which can be used with policy, in a policy context, and some simple fact sheets as well. We're starting at a much lower base than you are in North America. We published a bunch of different articles now, and including Sean published, and he's too modest to put it up, but an evaluation of potential indicators of temporal sampling protocols for monitoring genetic erosion. As I said, back in September in 2014, we were given permission to establish a genetic specialist group within the IUCN for the first time. We're currently upscaling, and we're already dealing with a whole variety of quite interesting problems, including the one of intentional genetic manipulation in southern Africa, where there is deliberate breeding of phenotypic variants to the exclusion of the natural populations, because these attain a very high financial value, millions of rand, for instance. But we're very concerned about what the potential outcome could that be for natural gene pools. And then the other big thing that we're currently dealing with is something called the Nagoya Protocol, which is another CBD article, which is effectively for genetics studies and genetic research, is resulting in almost a complete slowdown to stop of movement of genetic material between countries. So a variety of different issues around that. So here's my final slide. We need to make progress towards metrics, but let's not confuse them with indicators. Those are two different things, and we have to be aware of that. Indicators will necessarily, I promise you, have to be highly derived. They may be simple categorical things, okay? We need to think about temporal change, but also spatial dimensions as well, especially with issues to do with key biodiversity areas. Ideally, they should be future-proofed. And so, the more that we can consider the shape of genetic data in coming years, the better, really. But we also have to account for this incredible diversity of legacy data sets that we're going to have to work with. And how are we going to compare a whole genome estimate of a particular parameter with an AFLP estimate of a particular parameter with an Alzheimer estimate of a particular parameter? The ultimate product has to be, from a policy perspective, easy to understand, regardless of the complexity at the back end. And it's going to be really enjoyable to discuss the complexity at the back end over the next few days. But please don't forget, ultimately, if we want to get on the table, we need to have indicators which are going to be relatively easy for anybody to understand.